Welcome back to my channel, dear students. In this video, I am going to tell you something about biological practicals. So, today is 5th September and the special day in which each and every student will be waiting for this day. And that day is Teachers' Day. Happy Teachers' Day to all my dear teachers. Let me share some words with you. I have a wonderful shairi which will all teachers will like, and that is फूल हो गुलाब जैसे महल हो ताज जैसे फूल हो गुलाब जैसे महल हो ताज जैसे और जिंदगी के हर कदम पर टीचर्स हो आप जैसे. So I have a thought for all my dear teachers, and that is teachers lead like lions, not by forcing, but by inspiring courage, discipline, and with a confidence. Yes. So, as I saw in many of the schools, colleges, some students will be there who will be not at all respecting their teachers. That is really very bad. And for those students, I am going to tell something. Until and unless you respect your teachers, first you have to respect your parents, because parents are the first teachers and the second parents are your teachers. Right? Your parents are first teachers and your teachers are second parents. Keep it in your mind. Until and unless you respect your teachers, you are never going to succeed in your life. Mark it. Highlight this point. Until and unless you respect your teachers, you are never going to succeed in your life. Because some hard-working students will also be there who will be thinking, I will be doing very much hard work and why I'm not succeeding in my life. So for each and everything, tiny small thing also there will be a reason. Maybe the God has planned something else more than you thinking, right? So dear students, always respect your teachers. There is nothing much more greater than parents or the teachers, because friends will be coming and going in your life, but the teacher's blessing and your parents' blessing which is there, it will be forever in your life until and unless you succeed in your life. So always respect your teachers. And once again, happy Teachers' Day to all my dear teachers. So let me start with the biological practicals. I'm starting with the PUC second year syllabus. So PUC second year syllabus, in that I have took the topic known as biological practicals, right? And there are something 19 to 20 topics which you will be learning in the biology practicals. So I'll just tell you about the topics which all will be there. So the first is reproductive parts of hibiscus flower. This is the main question which you will be having in your exam, practicals exam. And pollen grains germination, pollen tube growth on stigma is also very important and it will definitely come in your exam. And obviously it is for something 5 to 10 marks. So you have to do it. Do the experiments with very carefully. Staining of nucleic acid, gametogenesis, common disease causing organisms and symptoms of the disease. Blastula of mammals, study of mitosis in onion root tip. Even this is the main question. Analysis of pedigree chart which is there, it will be there for your theory exams also and even the practical exams also. And the study of homologous and analogous organs, even that is for theory and practical. Living organisms in water samples, study of meiosis using permanent slides. Study of mitosis of onion peel. This is also very important. Female gametophyte development, and to verify law of segregation. To verify the Mendel's law of independent assortment. To perform emasculation, bagging and tagging for controlled pollution. Pollination. To study plant population density by quadrant method. And to study plant population by quadrant method. Yeah, there are 2 times repeatedly. So if I start now telling about the reproductive parts of hibiscus, you have to draw neat label diagram of hibiscus and label the parts very carefully, very neatly, because it consists something 5 marks in the theory also, even in the practical. Then you have to write some of the points regarding that, like androecium, gynoecium, what is flower, and you have to do the transfer section of hibiscus ovary. And you have to write about aim, principle, procedure, observations. These 4 points which are there, I will repeat once again. Principle, aim, procedure and observation with the diagram you have to consider in each and every experiments. Right? So, the diagrams are very important in your exam, even in the theory and practicals also. So, please don't try to skip your diagrams in any of the condition, because from the diagrams only you will get the passing marks. But you have to score in the exam, not just pass. Passing is easy, but failing in the exam is very difficult. Highlight this point also. And then you will be having pollen grain germination. And this is really very simple and easy experiment which you can do very well. So in this also, aim, principle, requirements, procedure and calculation part is also present in this experiment. Okay. So yeah, let me start with the first topic in the biology practical, and that is reproductive parts of hibiscus flower. And the parts which I am going to mark in this diagram, just listen it carefully. So this is the diagram of hibiscus flower. So you have to draw very neat, very carefully, because if you miss any one part also for labeling, you will obviously lose the marks. Let me start. This upper part which is there, look carefully. It is known as stigma. Okay, that is known as stigma. And this which you see in the hibiscus flower, whenever you will be studying of the biological experiments, you need to have the objects in your hands. Basically now I am not having, so I am doing it on the board, explaining it on the board. This part which is there of the hibiscus, it is known as stamens. The small parts which you can see, I'll just zoom out. These small parts which you can see, those are known as stamens. And the long part which you can see, this long part, middle part which is there, it is known as staminal tube, right? And then this all are petals. This which you can see, it is known as petals. Then this round part in the diagram in between the petals which is there, it is known as ovary. This is the main part which is going to play a huge role in your syllabus, first chapter. And then this part which you can see, it is known as thalamus. And this long root which you will be catching in your hand, the hibiscus flower, that is known as pedicel. So you need to explain about each and every part from this flower now, in which it is already present in your NCERT PUC second year textbook. It is in very detailed, it is explained very detailed, right? So let me go for the next section. So even you will be having the figure androecium, which is known as stamen, and that diagram is also very important. It is asked in MCQs also. No matter if it is not asked in 5 marks or 10 marks, but definitely it will be asked in MCQs or fill in the blanks with the diagrams. Right? So now it is androecium, which is also known as stamen. Right? So the stamen will be something like this. You need to simply draw like this. This mushroom like a structure which you can see is known as stamen, and this is known as anther, and this will be filament. It is asked in MCQs or in the fill in the blanks, or even in the 5 marks questions which will be there. It will be not exactly 5 marks. Some cutted questions will be there like for 3 marks, 1 marks and 1 marks for each question. So in that also you will be asked. And after this androecium, the gynoecium is also present. And in gynoecium, there is also a simple diagram which I have showed you in the hibiscus flower. The pistil is also main. Gynoecium, which is known as pistil. So this which is there, it is represented as female flower. And the androecium which was there, it was male. So I'll just draw this. So as I was told you as a last diagram, this is known as stigma, and the stamens will be present in that. And yeah, this long tube-like structure which you are seeing, it is known as style. Right now, for this two labelings, you will get 2 marks, and the diagram is not full. I'll draw it, and this diagram will be for 1 mark. This is known as pedicel, ovary, right? Yeah. And one more way is thalamus. So after drawing full diagram with the exact labeling parts, you are going to get 2 to 3 marks. So I'll just read for you the definition of the flower. Listen carefully, okay? So flower, what is flower? Flower is a modified shoot meant for sexual representation, reproduction, not this thing. So I'll just read for you the definition of the flower. So the flower is a modified shoot meant for sexual reproduction. The 4 flowers of a bisexual flower are calyx, corolla, androecium and gynoecium. So the androecium is the male reproductive organ of the flower, as I said in the diagram itself. The unit of androecium are stamens. Each stamen has 2 parts, namely anther and filament. Anther is sac-like structure which contains pollen grains or pore, and the pollen grains produce male gametes. The mature anther breaks and releases the pollen grains. The stalk of anther is called as filament. So you might get answer like what is filament, then you have to mention there the stalk of anther is called as filament. And then the pollen grains are transported to the stigma by the process called pollination. The question will also come like what is pollination? The pollination is the transformation of the stigma by the process. The pollen grains which is there, transformation of the pollen grains transported to the stigma market. It is known as pollination. So coming to the gynoecium, it is a female reproductive organ of the flower. The unit of gynoecium are pistil or carpels. Each pistil is composed of 3 parts, namely stigma, style and ovary. Ovary contains ovules, obviously. The role which is there of attachment of ovules to the ovary by placenta is known as placentation. Here also the question arises like what is placentation? So the definition will be the role of attachment of ovules to the ovary by the placenta is known as placentation. Now let us study the transverse section of hibiscus ovary. As I already showed you in the diagram, the TS it is known as. The full form of the TS is transverse section. Now let us come to the aim. It is the main point. So the aim will be to study of TS of hibiscus ovary by using a simple microscope. This the TS transverse section which is there, you will see it on the simple microscope, not in the compound microscope. Okay. So the procedure which is there, dissect a hibiscus flower and remove the ovary. So first, as this is the ovary, which you can see, here the petals are present, right? So first you have to remove all the petals. If this is a flower which I am handling it, you have to remove the petals first. Then inside you will get this part. If you remove these petals from the flower, inside you are going to get this type of diagram, means the structure. And then you have to just remove the filaments also from the flower. After removing the filaments, you will just get the ovary. Now you got this, right? You will cut it with the blade, which you have to handle it very safely. You have to remove the filament of the ovary, very thin shaped, very thin shape. It will be just like transparent. When you cut it with a blade, you will get the shape something like this. You have to continuously cut it with a spade, but with very carefully, okay? When you cut the filament of this ovary section, you will just keep it on the slide, the slide which will be there, the rectangular shape, you have to keep on this. I'll just show you. This is the part which you have cutted from the ovary. Then you will keep it on the slide. Okay. After keeping it on the slide, after taking the thin transverse section of the ovary and place them on the slide, you have placed them now. Now add 1 to 2 drops of water on the section of place a cover slip. Now listen very carefully. Whenever you are taking 2 drops of water on the slide, that time air bubbles should never enter into them. Because whenever you are covering with the cover slide, your cover slide also will be present. First of all, you will drop some amount of 2 drops of water. After dropping 2 times, then you will put a cover slip on that, on the cover slide. Remember, highlight the point, the air bubbles should not enter the cover slide. If it enters, then over. You will not get the correct information about that on the slide. Then, the slide is now ready for microscope observation. Now after putting 2 drops of water, you will cover with the cover slide. Then you will observe it in the microscope. Okay. Now we are going to observe it on the microscope. When you observe it on the microscope, you will get to know. Now what all you will see after keeping under the microscope. Observation point. Number of locules of ovary section. Now what is this locules? So in this diagram, some like this locules will be present. After looking it in the microscope only, you will get to understand very clearly. if this is a circle, the section of the ovary, you will get something structure like this. And the locules which are there, this, like for best example is pizza which is there. You will see the triangular shape, right? Like this is one locule. Okay, so they will be total 5 locules in the ovary section. And the type of placentation which is there, it is axial placentation, and this axial placentation you will be learned very well in the class 11th. The topic will be the morphology in the flowering plants. First you have to do your best in class 11 in each and every subject, not only biology, with clear diagrams, information and all. After then only you will be knowing very well of the class 12th. So these are the locules. So the question might come in your practical exam, the number of locules in ovary section equal to dash, and it will be 5. It depends on the flower. If it is 6, you have to write the 6. Then the type of placentation will be the question in your practical. So the type of placentation will be axial placentation, right? So this was your first practical, the reproductive parts of hibiscus plant. And then the hibiscus ovary, the diagram is also there. I'll just show you. It is also in the shape of circle only. So you have to draw neat label diagram of circle. In the circle, what you will label. So in between you have to make a small circle. In that you have to make a star-like structure like this, right? And then wherever the sharp edge is there, there you have to draw petals-like structure. In this you will be having the seed-like structure in between this. You have to draw really very neat. And this which I have said, those are locules in which the question is going to come. And this which the small seed-like structure which you are seeing, those are known as ovules. Then this central circle which is there, it is known as central part, central axis. Not part, it is known as central axis. And the last is placenta. The placenta you will be seeing in between the seeds, the space which is present in them, it is known as placenta. So this was all about your first practical in which you have to do it in your exam. So all the best, dear students. Do your best in your practical exams. Thank you.